unavoidable point of failure. Human eyesight cannot reliably resolve microscopic pathology. To fix this, researchers asked what would happen if you could equip a general practitioner.
For decades, automated skin cancer detection relied on computer-aided detection, or CAD. CAD is a legacy software framework that identifies disease based on rigid, manually programmed visual rules. Because human skin is visually inconsistent, these hard-coded instructions often failed to identify malignant tissue that didn't perfectly match the programmer's definitions. That approach reached a limit in 2017, when a team at Stanford University shifted the methodology. Instead of trying to write better rules, they designed a system that could learn the patterns of disease on its own. The researchers utilized deep learning, a specific type of machine learning modeled after the human brain, to process 129,450 clinical images of skin lesions. This massive dataset allowed the system to independently identify microscopic markers of cancer that are often invisible to the naked eye. To test the system, the team organized a benchmark study against 21 board-certified dermatologists. Both the humans and the machine were tasked with a binary classification challenge: look at a lesion and determine if it was malignant or benign. This graph charts the resulting diagnostic accuracy. The data, published in the journal Nature, showed the algorithm matching the experts with an area under the curve, or AUC, score of 0.94 to 0.96. AUC is a statistical measurement of how effectively a test can distinguish between two distinct groups, such as healthy and cancerous cells. The results proved that clinical intuition was no longer the only way to identify a melanoma. A computer had reached the same tier of accuracy as a human specialist. AI learns through sheer volume and repetition, similar to how a child learns to tell a cat from a dog. You don't provide a child with a list of mathematical rules. You show them thousands of examples until the brain recognizes the pattern instantly. The architecture behind this is the convolutional neural network, or CNN. A CNN is a type of deep learning model that simulates biological neurons to process images and extract complex visual patterns. Most medical models use transfer learning, which is the process of taking an AI already trained to recognize basic objects and fine-tuning it with a specialized dataset. Developers often use the ISIC archive, which contains over 10,000 high-resolution images of skin lesions. ISIC is the International Skin Imaging Collaboration, a global open-source project designed to improve digital dermatology. This radar chart shows how a trained CNN performed in a real-world benchmark against 96 dermatologists. The AI achieved a 95% sensitivity and an AUC of 0.918, effectively matching the performance boundaries of the human specialists. However, high accuracy often depends on a dermatoscope. This tool uses polarized light and high-powered magnification to create uniform, detailed images. Standard smartphone photos are much harder for a machine to interpret because they are often out of focus or poorly lit. Deep learning systems succeed because they don't rely on human definitions. They scan millions of pixels to invent their own mathematical markers for identifying disease within the chaos of a clinical photograph. The diagnostic pipeline begins the moment a lesion is captured by a lens. First is pre-processing. Algorithmic masks sweep the image, digitally erasing artifacts like hair or blood vessels. Next, a Gaussian filter blurs the image slightly, reducing digital noise and normalizing raw pixel values into a uniform grid. The cleaned image then enters the CNN. Early layers scan for simple edge features, while the deeper layers identify complex, irregular textures indicating a malignancy. This is feature extraction. The network condenses thousands of details into abstract mathematical weights. In the scoring phase, these millions of adjusted weights collapse into a single probability calculation. The final output of the pipeline is a binary prediction, malignant or benign, presented with a specific percentage of confidence. This entire process occurs almost instantly, a fatigue-free mathematical analysis that operates far faster than a human clinician can physically examine a patient. This technology has already moved out of the lab and into the real world. Clinical-grade systems are already in use. Doctors are currently using FDA-cleared hardware like DermiSensor, as well as European-approved software like Molinizer Pro to help them make bedside decisions. Consumer-facing applications like SkinVision and FirstDerm have also launched, putting this diagnostic pipeline directly onto a patient's smartphone. But these impressive accuracy rates hide several critical flaws that the data is only beginning to reveal. This chart shows the racial performance gap identified in a 2022 Stanford study. AI performance measured in 0.89 AU-ROC on light skin, but dropped to 0.82 for patients with darker skin tones, categorized as Fitzpatrick 4 through 6. The Fitzpatrick scale is a numerical system to classify human skin color based on its response to ultraviolet light. Beyond bias, these black box mobile apps are also vulnerable to physical adversarial camera attacks. An adversarial attack is a technique that uses imperceptible visual perturbations to trick an AI into making a false diagnosis. AI can only be as objective as the images we use to train it. Without diverse datasets and robust security, we risk turning a powerful medical tool into a source of racial inequity and digital risk.
We have seen how early algorithms stumbled, but developers are actively rewriting the code to fix dataset bias and build equitable healthcare tools. The foundation of this fix starts with the data itself. Researchers built the Fitzpatrick 17K dataset. This image library is explicitly designed to balance training data evenly across all human skin tones. Legacy AI models showed a massive 27% sensitivity gap for darker Fitzpatrick skin types. By utilizing generative AI to synthesize training images of melanomas on darker skin, researchers mathematically equalized the algorithm's performance across all skin tones. Correcting this baseline data is a mandatory first step. If we deploy these tools without balancing the scales, we risk baking current health disparities directly into the algorithms of the future. Even with perfect data, doctors still face the black box problem, an algorithm that spits out a diagnosis but cannot explain its reasoning. That is why the field is actively shifting toward explainable AI. This graphic shows a vision transformer generating a self-attention map. These deep learning models process images by dividing them into distinct patches. The resulting heat map highlights the exact pixel boundaries of a lesion, showing precisely where the AI focuses. By mapping out its visual thought process, the AI stops acting like a mysterious, unquestionable oracle. It becomes an accountable second opinion that a doctor can actually verify. When a patient's life is on the line, physicians cannot blindly trust a machine's output. They require the AI to show its work. Today, dermatologists generally agree on how this technology should be applied. AI functions as an expert guide and a validator to support clinical decisions, rather than an autonomous replacement for human judgment. We are seeing this dynamic in practice with the DermiSensor. This is the first FDA-cleared AI breakthrough currently operating right now in primary care clinics. This diagram illustrates how the device utilizes elastic scattering spectroscopy. Elastic scattering spectroscopy is an optical technique that bounces light waves into the cellular structures of the skin. Those waves bounce off irregular cellular structures and scatter back to a collection sensor, gathering real-time data on the tissue. As you can see in this data readout, giving a primary care physician this tool creates a sharp upward trend line in their performance. With the device, their diagnostic sensitivity in identifying malignant lesions jumps to over 91%. The resulting impact on patient outcomes is massive. By augmenting the physician's exam with optical data, primary care clinics see a bar plummeting by 68% for missed skin cancers. This same collaborative approach is improving teledermatology networks. When rural clinics use AI assistants to review remote cases, their diagnostic agreement with top specialists increases by up to 12%. AI's greatest immediate value lies in bringing specialist-level accuracy to the front lines of primary care, bridging the geographic access gap for millions of patients. Looking ahead, these point-of-care spectroscopy devices will continue to shrink until they are as ubiquitous and miniaturized as the modern stethoscope. At the same time, consumer smartphone apps will achieve clinical-grade accuracy through transfer learning. Transfer learning is a machine learning technique that takes a model trained on high-quality clinical data and adapts it to work accurately with noisy, lower-resolution camera hardware. The next major leap in the medium term is multimodal AI. Multimodal AI is an algorithm capable of processing multiple different types of data, like images, text, and genetics, simultaneously. This model shows how multimodal AI works. It combines three distinct data streams: the visual node of a skin scan, the text node of a medical history, and a DNA helix representing genomics, merging all of it into a single, forward-moving predictive timeline. With this technology, the diagnostic timeline shifts entirely. We move from identifying a cancer that already exists to predicting its formation long before it becomes a threat. Fast forward to 2035. Picture a teenager in rural India holding a smartphone over their arm. Using an app powered by decades of refined data, they receive an instant, highly accurate, and free diagnosis right there in the glow of the screen. Compare that immediacy with our current medical system, where patients endure agonizing months of waiting, anxiety, and expensive referrals just to get a specialist to look at a single mole. Think back to the patient we met at the start of this story. When evaluated instantly by a doctor using a handheld AI device, their months of uncertainty disappear, replaced by immediate clarity and an early, highly treatable diagnosis. The central question of this technological race is whether we have the courage and the discipline to ensure these tools are built to serve everyone equally. The future of medicine relies on artificial intelligence, but it will only become truly human through the choices we make today.
If you're enjoying this, subscribe. It really helps the channel.
Artificial intelligence is rapidly entering dermatology clinics, promising to catch skin cancer earlier and with greater accuracy than human doctors. But buried in the code of these new diagnostic tools is a fatal flaw, one that directly targets patients with darker skin. To understand the risk, we have to look at the clinical baseline. This chart compares melanoma outcomes by race. While skin cancer is less common in Black patients, they present with late-stage disease 52% of the time. Because it is caught so late, their 5-year survival rate drops to 68%, compared to 92% for white patients. A major driver of this disparity is a specific subtype called acrolentiginous melanoma. It frequently appears on the palms of the hands or the soles of the feet, areas that are easily overlooked during routine exams, especially on darker skin. Computer vision algorithms were proposed as the perfect objective tool to close this survival gap. They don't get tired, and they don't rush through physical exams. Yet, current implementations are actively exacerbating the divide. The issue originates in how these models learn. Machine learning requires massive volumes of ground truth training data to recognize patterns. If the data is skewed, the resulting medical tool will be too. This pie chart breaks down the demographic makeup of HAM 10,000, which is a widely used dataset of 10,000 training images for skin lesion classification. Exactly 81% of the images in this database represent light skin types, classified as types I through 3 on the Fitzpatrick scale, the standard classification system for human skin color. The imbalance repeats across other major databases. When looking at the ISIG archive, the International Skin Imaging Collaboration, only 9% of the images represent the darkest skin tones, Fitzpatrick types 5 and 6. Neural networks learn to detect the borders, colors, and textures of malignancies based strictly on what they are fed. By training them almost exclusively on white skin, developers have built tools that cannot reliably decipher the visual signatures of cancer on diverse populations. An algorithm is a perfect mirror of its inputs. It does not create bias out of thin air. It mathematically scales the blind spots of the medical system that built it. We can quantify exactly how dangerous this data gap is. In 2022, Dr. Roxana Dineshju and a team at Stanford University tested several major AI dermatology systems using clinically validated images representing all skin tones. This chart maps the algorithm's sensitivity, its ability to correctly identify a malignant lesion. For patients with very light skin, the AI performed exceptionally well, successfully detecting 92% of the melanomas. But when the researchers ran images of dark skin tones through the exact same system, the sensitivity plummeted to 65%. In clinical terms, out of 100 melanomas in dark skin patients, the AI entirely misses 35 of them. That is 35 aggressive cancers ignored by the machine. A physician leaning on the software's high overall accuracy rating might send a patient home without a biopsy. Deploying these models prematurely provides a false sense of security that is actively dangerous for patients of color. Compounding the high failure rate is the black box problem. Neural networks cannot explain their logic. When the system classifies a lethal lesion as benign, it cannot tell the doctor which pixels led to that conclusion. This creates a liability vacuum. If a doctor relies on an opaque, algorithmically generated recommendation and a patient dies from a misdiagnosis, the legal and ethical responsibility remains completely undefined in modern medicine. These systems are also technically fragile. Research shows they are vulnerable to adversarial attacks, a technique where tiny physical manipulations, like placing a specially printed sticker near a lesion, can confuse the camera and force the AI to return a false diagnosis. There is also a quiet harvesting of data occurring in the background. Many tech companies absorb patient images to train future iterations of their software, often embedding broad consent clauses in their terms of service to monetize the data later. Patients in under-resourced communities frequently turn to digital tools out of necessity. They end up providing the raw data used to refine these algorithms, only to be priced out of the final commercialized medical device. While clinical tools require some oversight, millions of patients are downloading unregulated, direct-to-consumer smartphone apps that promise to assess skin cancer risk right from their living rooms. This visualization maps the diagnostic accuracy of these consumer apps. Sensitivity rates fluctuate wildly, ranging from 73% down to a useless 7%. In one Dutch study, SkinVision flagged 19% of lesions as high risk, but dermatologists found only 0.7% were actually abnormal. This severe overdetection creates panic. It subjects healthy people to emotional distress and drives thousands of unnecessary biopsies, clogging up waitlists for patients who actually need urgent dermatological care. The FDA vets medical technology, but there's a massive legal distinction. FDA approval requires rigorous clinical trials. FDA clearance only requires showing a device is substantially similar to an existing one. Developers exploit this gray area, avoiding rigorous physician-level validation. Artificial intelligence possesses the computing power to revolutionize dermatology, but its current trajectory is deeply flawed. The underlying data is biased, the consumer tools are dangerously unregulated, and the algorithms fail the exact populations that need early detection the most. We have the technology to map the human body down to the pixel. The question is whether we will build a tool for universal health or code our existing disparities permanently into the future.
Inside a quiet examination room, a doctor leans in, adjusting a glass lens over a jagged, dark blemish on a patient's arm. The physician hesitates, weighing the microscopic variations in color and shape. Right now, millions of people around the world are waiting in rooms exactly like this one. They rely on these highly subjective visual assessments to tell them if they are healthy or if a deadly cluster of cells has begun to spread. When a doctor successfully identifies a melanoma at 1 millimeter thick or less, the 5-year survival rate exceeds 97%. But if it grows just a fraction thicker, advancing to stage 3, survival falls between 36 and 63%. Survival in these cases depends almost entirely on catching tiny, fractional changes on the surface of the skin. It is a geometry problem that the naked human eye struggles to solve with consistent accuracy. Decades ago, spending long hours in the sun without protection became a normal part of recreational culture. That mid-century embrace of tanning laid the biological groundwork for a massive spike in cellular damage that is surfacing today. The result is a global health crisis of staggering proportions. Skin cancer now accounts for at least 40% of all cancer cases diagnosed worldwide. The damage is highly concentrated in specific regions. Cities like Auckland, New Zealand, and population centers across Australia report the highest incidence and mortality rates on Earth, driven by heavy ultraviolet exposure and a predominantly light-skinned population. This rising bar chart projects the trajectory of the disease in the United States over the coming decades. By the year 2040, cases are expected to reach 219,000, making melanoma the second most common cancer in the country. Millions of new patients require immediate, specialized attention every year. The physical volume of the disease has grown so large that it has overwhelmed the medical infrastructure built to treat it. Across the globe, healthcare systems are hitting a hard mathematical bottleneck. There are simply not enough trained dermatologists to examine everyone who needs a screening. This ratio compares specialists against the population. France has 4.4 private practice dermatologists per 100,000 people. The US drops to 3.66. In low and middle income countries, infrastructure practically disappears. Entire regions operate with fewer than one specialist per 100,000, leaving massive populations without access. This scarcity forces patients to cross immense physical distances just to be seen. A farmer in rural Queensland might have to drive 400 kilometers across the outback for a single 15-minute skin check. Even in densely populated areas, time is a severe barrier. The average wait for a dermatologist appointment in places like France spans 95 days. During those 3 agonizing months, an undetected tumor continues to divide and grow much deeper into the tissue. These diagnostic delays carry an immense financial burden. Health economists track this using Quality Adjusted Life Years, or QALY, which is a metric that measures the value of health outcomes by combining the length and quality of a person's life. Interventions to catch skin cancer can cost healthcare systems up to $80,000 per QALY gained. A patient's physical distance from a clinic and the month spent waiting on a schedule frequently decide their fate. In many cases, these logistical barriers prove more lethal than the initial cellular mutation itself. Yet, when a patient finally clears the waiting list and sits on the examination table, the actual diagnostic process remains heavily subjective. This diagram visualizes the ABCDE rule, the standard criteria doctors use to evaluate moles. They look for asymmetry, border irregularity, color variations, diameter over 6 millimeters, and evolution, or how the lesion changes over time. Because humans are applying this filter manually, they naturally err on the side of caution. Doctors frequently flag harmless shapes as suspicious, leading to massive overdiagnosis of benign lesions. This cautious approach triggers unnecessary biopsies, wastes clinical resources, and causes severe psychological distress for the patient. Conversely, relying strictly on these visual rules leads to fatal blind spots. Rare and aggressive variants, like amelanocytic melanomas, which are dangerous skin cancers that lack the typical dark pigment, often bypass the criteria entirely. No matter how many years of medical school a specialist completes, the human eye has hard biological limits. It is an imperfect tool trying to manage a disease that demands microscopic precision. The medical community is hitting the upper limits of what human vision and manual scheduling can achieve in the fight against melanoma. To catch these cellular anomalies before they become lethal, the point of diagnosis has to move out of the specialist's office. It has to move closer to the patient. Computer scientists are now training artificial intelligence systems to analyze skin lesions. They are asking if an algorithmic lens can accurately spot the microscopic geometric patterns that humans routinely miss. The most effective defense against the world's most common cancer may not require building more hospitals or waiting 3 months for an appointment. The solution may already be sitting in your pocket.
Quick pause. Did you know only 20% of you watching are actually subscribed? If this content
Inside a typical primary care clinic, doctors often have about 15 minutes to examine a patient, evaluate symptoms, and decide if a suspicious mole could be fatal. The biological reality is that at stage 0, a microscopic cluster of melanoma cells looks exactly like a harmless freckle. The human eye simply lacks the physical resolution needed to see the microscopic cellular structures that define early-stage cancer. This chart compares the actual accuracy of primary care providers against a perfect 100% detection rate. Statistically, general practitioners correctly flag about 83% of melanomas prior to a biopsy. That leaves a 17% gap. That margin of error creates a massive systemic burden. Thousands of patients undergo unnecessary biopsies for benign moles, overloading dermatology referral networks with long waitlists. At the same time, aggressive cancers are inadvertently sent home with a clean bill of health. The global pipeline for diagnosing skin cancer is bottlenecked by a single, unavoidable point of failure. Human eyesight cannot reliably resolve microscopic pathology. To fix this, researchers asked what would happen if you could equip a general practitioner with the optical resolution of a specialist right inside the exam room. The answer lies in a technology called elastic scattering spectroscopy, or ESS. ESS is an optical sampling technique that uses light to read the microscopic structure of living tissue without having to cut it out of the body. Improving early detection rates requires doctors to stop relying strictly on human vision and adopt a new physical method for capturing light and assessing tissue. This animation illustrates the physical process of ESS. A handheld device fires brief pulses of light into the skin. As those photons hit the cells beneath the surface, they scatter and bounce back in specific patterns. Healthy, orderly cells reflect light evenly. But malignant cells are structurally chaotic. They have larger nuclei and irregular boundaries, which forces the photons to scatter into a highly erratic optical signature. A machine learning algorithm then analyzes that exact scattering pattern. The software was trained on over 10,000 spectral recordings of known skin lesions. By comparing the live scan to its database, the AI calculates the risk level and returns a simple instruction for the doctor: monitor or investigate further. The device bypasses human visual limitations entirely by measuring the physical interaction between light and cellular geometry. To test this in real-world conditions, researchers ran two major clinical trials, DermSuccess and DermAssess-3. They tested an ESS device called DermiSensor directly against the diagnoses of primary care physicians. Looking at this chart comparing the trial results, the physicians correctly identified 83% of the cancers. The ESS device detected 95.5% of them. The data also highlights a metric called negative predictive value, or NPV. NPV measures how likely a negative test result is actually correct. The device achieved a 96.6% NPV, meaning if the machine says a mole is fine, there is only a 3.4% chance that it is malignant. Those clinical trial results provided the objective evidence needed for the device to be cleared by the FDA for use in the United States. The medical community now has regulated data proving that an algorithmic optical tool can accurately identify malignant tissue that general practitioners miss. Algorithms are only as reliable as the data they learn from. If a machine encounters a biological profile it wasn't extensively trained on, the underlying math breaks down. A 2022 study out of Stanford University analyzed how multiple artificial intelligence models for dermatology performed across a diverse set of patients. The researchers found a severe imbalance in the training data. Most algorithms were overwhelmingly fed images of lighter skin, classified in dermatology as Fitzpatrick skin types 1 through 3. The Fitzpatrick scale is a numerical system used to classify human skin color based on its reaction to sunlight. This graph maps AI diagnostic accuracy against the Fitzpatrick scale. While the models easily identify cancers on pale skin, their accuracy drops significantly when evaluating darker skin tones, types 4 through 6. Relying on these uncorrected models means patients with darker skin are mathematically more likely to receive false negatives, turning existing healthcare inequalities into automated features of the system. While clinical hardware faces strict oversight and trials, a parallel industry is putting unverified algorithms directly into the hands of patients via consumer smartphone apps. Many of these apps bypass the FDA entirely. Instead, they operate under regulations like the European Medical Device Directive, which allows developers to effectively self-certify their software as long as they include a basic medical disclaimer. A major review in the British Medical Journal tested these apps. They found that variations in smartphone camera quality, poor lighting conditions, and inconsistent user technique cause immense diagnostic inaccuracies, leading to false security and rampant overdetection of benign moles. Machine learning offers a powerful method for decoding the physical structures of disease, provided it is rigorously tested and assists a trained professional. If you are concerned about a lesion, skip the App Store, see a physician, and advocate for diagnostic tools that have been proven to work accurately on every shade of human skin. Millions of people are downloading artificial intelligence apps to their smartphones to scan their moles for skin cancer. But systematic reviews of these applications, including a major meta-analysis published in the British Medical Journal, show their performance is highly inconsistent. The clinical data reveals that some of the most popular apps incorrectly classify 30% or more of actual melanomas as completely benign, while simultaneously flagging harmless lesions as high risk. The underlying problem with these applications is not the code powering their AI algorithms. The failure stems from the physical limitations of the smartphone camera hardware capturing the images. Any artificial intelligence is only as reliable as the physical sensor feeding it data, and a smartphone camera is physically blind to the cellular markers of cancer. To solve this problem, the U.S. Food and Drug Administration recently cleared a new category of medical device built specifically to bypass the flaws of smartphone cameras. This is DermiSensor, a new hardware class designed for primary care physicians. It completely abandons two-dimensional photography. Instead, it relies on elastic scattering spectroscopy, or ESS, an optical technique distinguishing normal from abnormal tissue by recording how photons scatter. To reliably detect malignant tissue, diagnostic tools must stop analyzing the superficial color of the skin and start measuring its physical, subcellular structure. Smartphones capture surface light as a 2D grid of RGB pixels. Ambient light temperature corrupts these values before AI processing. Internal white balance filters further distort this diagnostic data. Finally, natural melanin density confounds the readings, creating systemic bias against darker skin tones. Smartphone AI fails because it is attempting to diagnose a deeply structural, cellular disease using easily corrupted surface reflections. ESS functions as an optical biopsy, extracting structural data by firing light into the tissue and analyzing the reflection, rather than surgically cutting the skin. The device emits broadband light, measuring how photons scatter against cellular boundaries. Normal nuclei reflect predictably. Enlarged malignant nuclei force photons into chaotic paths, altering the optical signature. By measuring physical geometry, the spectral waveform remains mathematically identical across all skin tones. ESS physically bypasses the optical illusions of color. It uses physical geometry to calculate the presence of malignant cellular structures. The definitive clinical test for this physics-based approach was the DermSuccess pivotal trial, led by researchers at the Mayo Clinic. The study was conducted across 22 clinical centers, testing over 1,500 suspicious lesions on more than 1,000 patients. It achieved 96% overall sensitivity for detecting skin cancers, 88% of melanomas, 98% of basal cell carcinomas, and 99% of squamous cell carcinomas. It also achieved a 97% negative predictive value, meaning a negative result is highly trustworthy. Clinical data objectively proves that measuring subcellular photon scattering is vastly superior to evaluating surface pixels when ruling out a malignancy. An independent study conducted at the University of Pittsburgh Medical Center explored the statistical trade-offs required to engineer a device that misses almost zero cancers. Setting the threshold to catch 100% of melanomas drops specificity to 9.4%, netting harmless lesions. Adjusting positive scores to 7 through 10 balanced the scale, bringing sensitivity to 86.4% and improving specificity to 67.2%. This intentional statistical imbalance is perfectly suited for a triage tool used by general practitioners, whose job is deciding whether a patient needs a specialist. A triage device doesn't need to be perfectly specific. It must be highly sensitive to ensure no deadly cancer ever slips through the cracks. Currently, primary care physicians often refer all suspicious lesions to dermatologists, creating massive wait times for specialist care. This chart shows how the ESS device creates a new workflow. Primary care doctors scan suspicious lesions immediately at the point of care, and the device generates a binary result: monitor or investigate further. Backed by the 97% negative predictive value, the monitor result gives the doctor the confidence to send the patient home, safely keeping them off the dermatology waitlist. Conversely, an investigate further result prioritizes high-risk patients, routing them directly to a dermatopathologist for a definitive physical biopsy. Arming primary care gatekeepers with subcellular optical tools allows them to catch cancer earlier while optimizing the entire healthcare pipeline. These ESS devices do not replace the dermatologist, and they do not replace the physical biopsy. They are an early-stage filter. There is an inherent danger in relying on unregulated pixel-based smartphone apps for medical triage. True clinical reliability requires purpose-built, FDA-cleared hardware. The tech industry's intense focus on software algorithms has distracted from the necessity of engineering better physical sensors to gather medical data. The future of diagnostic AI relies not on downloading better apps to consumer phones, but on building clinical sensors that view the human body through.
Thank you for exploring the future of medicine with us. If you found this valuable, subscribe to the channel and join the conversation in the comments below. See you in the next one.